Today we are going to discuss about various aspects of fermentation. Fermentation is a metabolic process that converts sugar to acids, gases and alcohol. It occurs in yeast, bacteria and in oxygen-starved muscle cells. The term fermentation is more broadly used to refer to the bulk growth of microorganisms on a growth medium to produce a specific chemical product. Fermentation process utilizes microorganisms to convert solid or liquid substrates in different products. French microbiologist Louis Pasteur has worked on fermentation and its microbial causes. The science of fermentation is known as zymology. The term fermentation is derived from the Latin word fervor, which means to boil. It describes the appearance of action of yeast on extracts of fruit or malted grain. The boiling appearance is due to the production of carbon dioxide bubbles caused by the anaerobic catabolism of the sugars present in the extract. In biochemistry, fermentation is related to the generation of energy by the catabolism of organic compounds. The catabolism of sugar is an oxidative process which results in the production of reduced pyridine nucleotides. These must be reoxidized for the process to continue. Under aerobic conditions, reoxidation of reduced pyrimidine nucleotide occurs by electron transfer via the cytochrome system, with oxygen acting as the terminal electron acceptor. However, under anaerobic conditions, reduced pyrimidine nucleotide oxidation is coupled with the reduction of an organic compound. This is often a subsequent product of catabolic pathway. During the action of yeast on fruit or grain extracts, NADH is generated by the reduction of pyruvic acid to ethanol. Different microbes are capable of reducing pyruvate to a wide range of end products. Thus, the term fermentation has been used in a strict biochemical sense to mean an energy generation process in which organic compounds act as both electron donors and terminal electron acceptors. The production of alcohol by the action of yeast on malt or fruit extracts has been carried out on a large scale for many years and was the first industrial process for the production of a microbial metabolite. Thus, industrial microbiologists have extended the term fermentation to describe any process for the production of a product by the mass culture of a microorganism. There are 5 major groups of commercially important fermentations. Production of microbial cells or biomass, for example, yeast, SCP. Production of microbial enzymes, for example, amylase, protease, lactase, glucose oxidase. Production of microbial metabolites, for example, citric acid, glutamic acid, lysine. Production of recombinant products, for example, interferon, insulin, human serum albumin. Modification of a compound which is added to the fermentation, for example, the transformation process like vinegar, steroids, etc. Fermentation is a natural process. People applied fermentation to make products such as wine, meat, cheese and beer long before the biochemical process was understood. Louis Pasteur studied the methods and processes involved in manufacturing of wine and beer. He found that fermentation of fruits and grains was brought about by microbes. By proper selection of microbes, a quality product can be produced. Pasteur suggested that the undesirable type of microbes might be removed by heating. This process is known as pasteurization. Yeast and certain bacteria perform ethanol fermentation where pyruvate from glucose metabolism is broken into ethanol and carbon dioxide. The net chemical equation for the production of ethanol from glucose is C6H12O6 glucose gives twice C2H5OH ethanol plus twice CO2 carbon dioxide. Ethanol fermentation is used in the production of beer, wine and bread. The pyruvate molecules from glucose metabolism, glycolysis, may be fermented into lactic acid. Lactic acid fermentation is used to convert lactose into lactic acid in yogurt production. It also occurs in animal muscles when the tissue requires energy at a faster rate than oxygen can be supplied. The equation for lactic acid production from glucose is C6H12O6 glucose gives twice CH3CHOH COOH lactic acid. The production of lactic acid from lactose and water may be summarized as C12H22O11 lactose plus H2O water gives 4 CH3CHOH COOH lactic acid. Industrial fermentation involves A. the organism, B. the medium, C. the equipment. The organism used for industrial fermentations should have some characteristics such as it should be high yielding strain, it should have stable biochemical characteristics, it should not produce undesirable substances, it should be easily cultivated on large scale. The medium must be cheap and readily available in large quantities. Nutrient containing waste material can also be used, for example, waste from dairy industry like whey and paper industry like sulfite waste liquor. A fermenter is a specially designed equipment or a vessel in which microbes are grown in their favorable conditions in fermentation industries to obtain their product in large amounts. The fermenter should have all the facilities needed for monitoring and controlling the fermentation parameters. In surface fermentation, the organism is grown on the surface of the liquid medium. Therefore, large surface area is required for more yields. Agitation is not performed. As a result, process control of fermentation is difficult. Oxygen transfer is insufficient since the organisms in the top layer will get more oxygen than the organisms in the lower layer. Danger of contamination is more. Homogeneity cannot be achieved in this process. It is also time consuming. For example, citric acid production. In submerged fermentation, the organism is grown in the entire liquid medium. Large tanks called fermenters are used. They can be either open or closed. Continuous agitation is performed. Thus, proper homogeneity is maintained. Maintenance of constant temperature and pH control is easy. Availability of oxygen is also properly maintained. It is mostly used in industries, for example, vinegar production. Solid state fermentation is also known as SSF. It involves the growth of microbes on moist solid particles. SSF mainly involves filamentous fungi. The substrates used in SSF processes are often products or byproducts of agricultural, forestry or food processing. The source of nutrients comes from within the particles, for example, soya sauce manufacturing. Fermentation can be carried out by three different methods: A. Batch fermentation, B. Fed batch fermentation, C. Continuous fermentation. In batch fermentation, the fermentation process is undertaken in batches. A large volume of nutrient medium is inoculated and the process is run for product formation. After completion, the product is harvested and the fermenter is washed and sterilized for subsequent batches. This is a closed system in which all the nutrients are initially added to the vessel and inoculated. In fed batch fermentation, substrate is added in increments as the fermentation progresses. In the beginning of the fermentation, the critical elements of the nutrient solution are added in small concentrations. Later, they are added in small doses during the production phase. Under the continuous fermentation process, fermentation takes place continuously. Components of the upstream processes are withdrawn intermittently and replaced with fresh media or nutrients. This is done through single stage fermentation and multiple stage fermentation methods. In the single stage fermentation method, a single fermenter is inoculated and then kept in continuous fermentation. The multiple stage fermentation method involves two or more stages with the use of two or more fermenters in sequence. In this, growth occurs in the first fermenter and synthesis in next fermenter. Process monitoring is done by turbidostat. Several factors can affect the fermentation process and the final quality of the product. Factors which are most important to control are temperature, pH, nature and composition of the medium, aeration or dissolved oxygen, agitation, foam control and operating systems. Temperature has an impact on the growth and activity of different types of microbes. Therefore, it should be maintained during the fermentative production. pH is also a key factor for fermentation. Sometimes a characteristic product is formed only at a particular pH. For example, during ethanol production, when the pH value is above 5.0, the quantity of ethanol produced substantially decreases. Therefore, a pH range of 4.0 to 5.0 may be regarded as optimum for the anaerobic ethanol production process. pH plays an important role in determining the fermentation pathway used in anaerobic ethanol production processes. The formulation of the medium affects the yield, rate and product type. The medium must provide the necessary amounts of carbon, nitrogen and trace elements and micronutrients. Aeration could be beneficial to the growth and performance of microbial cells by improving the mass transfer characteristics with respect to substrate, product and oxygen. Agitation is also an important parameter for adequate mixing, mass and heat transfer. Agitation creates shear forces causing morphological changes, variation in their growth and product formation and damage to the cell structure. The aeration and agitation of fermentation broths generates foam. Typically, 20 to 30% of the fermented volume must be left empty to accommodate the foam and allow the gas for disengagement. The type of operating system selected, whether batch, fed batch or continuous system, also affects the process of fermentation. Experimental fermentation conditions are determined in flask or small fermenters in the laboratory and then employed on a grand scale for operation of the large production fermenters. However, small to great differences occurs between the designs and efficiencies of small and large scale fermenters. This can bring about changes in the parameters of the fermentation process. Therefore, the process of scale up is undertaken. Scale up is the determination of the proper incubation conditions to be employed with the large scale production tanks based on information obtained with small tanks. Larger experimental tanks used for scaling up are called pilot plants. Various organisms are responsible for the production of varied products through fermentation. Bread, beer, wine and spirit are produced by Saccharomyces cerevisiae. Cheese is produced by Penicillium species. Fermented meats and vegetables are produced mostly by lactic acid bacteria. Soya sauce is produced by Aspergillus oryzae. Today we have discussed about fermentation, its various definitions, history and types, and various aspects of industrial fermentation, including its prerequisites, the various processes and operating systems needed for it, and some products that are commercially produced through this process. I hope